As we consider the processes of transcription and translation, we will see that the three main types of RNA involved are mRNA, tRNA, and rRNA. In order to function, each of these must be modified to a final form. We'll take a look at that process in this lesson. Each of these RNA molecules exists first in a nascent form, but must be processed in order to become functional. This process varies somewhat between prokaryotes and eukaryotes, and this is particularly true for messenger RNA. The types of modifications that occur include trimming of leader and trailer sequences, addition of sequences, and chemical modification of certain bases. In the next chapter, we will look more particularly at the role of transfer RNA, or tRNA. It is initially transcribed as one long single-stranded RNA molecule, but is subsequently trimmed. As shown below, an initial RNA contains what will become two separate tRNA molecules, one that carries the amino acid proline on the left, and the other, on the right, will carry serine. After initial trimming, a terminal sequence is added, and some of the bases are also chemically modified in order to reach its mature or final form. Each tRNA carries the CCA sequence at its 3' end. Ribosomal or rRNA also undergoes editing, including methylation of bases and trimming. There are 3 rRNA molecules that associate with proteins into the two subunits that comprise the prokaryotic ribosome. In eukaryotes, the dimeric ribosome has 4 separate rRNA molecules. The sizes vary between these organisms, though they share a common function. Chemical modification is primarily accomplished through methylation. The greatest difference in processing between prokaryotes and eukaryotes has to do with mRNA. Keep in mind that there are several types of RNA molecules in the cell, and only mRNA is targeted for translation into protein. The challenge is to properly identify an RNA molecule as a message to be translated. Eukaryotes accomplish this by modifying both the 5' and 3' ends of the message. A cap is added to the 5' end, as shown here, which is a methylated G residue. The nature of the linkage to the first nucleotide in the message is also distinct from that of the rest of the chain. Recall that the nucleotides are joined into the polymer as 5' to 3' linkages. However, the 5' cap is joined to the first nucleotide in a 5' to 5' linkage. This protects the message against nuclease digestion. The first 2 nucleotides in the message are methylated, which also helps to identify it as mRNA. Eukaryotic mRNA is also modified at the 3' end. A sequence of 100 to 200A residues are added in the nucleus, referred to as the polyadenylate or poly-A tail. This also protects the RNA from nuclease digestion and is needed for circularization in the process of translation, as we will see later. Eukaryotic mRNA is dramatically altered in another way, which we will look at in our next lesson.